Hi all, Dr. Clark here again. And for this lecture in Gen Bio, we're going to talk about gene expression. So we talked about transcription, so how are genes transcribed, and this was very basic. Basically, we said that the DNA is opened up, RNA polymerase comes in, makes a copy, the DNA closes back down, and you have yourself a messenger RNA. That is actually not quite true. And then we talked about translation, taking that messenger RNA and translating it into a protein. Well, there are many steps with gene expression in which you can regulate different processes. Different processes can be broken down. They can have negative or positive feedback loops. There's lots of situations. Proteins could be misfolded. Lots of situations where this can break down. We'll talk about that in a later lecture. But right now we're going to talk about how genes are actually expressed. What is the difference between prokaryotic expression and eukaryotic expression? So this is a diagram just showing some of those situations where the gene might be expressed differently depending on the organism, depending on the situation, in some cases depending on the age of the organism. So remember we talked about telomere length and we talked about the telomere caps. We talked about this in regards to Leonard Hayflick and then Elizabeth Blackburn discovering the Hayflick limit, or the reason for the Hayflick limit, and that telomeres get shorter and shorter after every division. This can change gene expression. Another way at which this can occur is you can have histone modifications. So you can get DNA that's has different tackness and different stickiness to the proteins that are associated with. You can get the methylization of DNA. You can get somatic mutations, which we've talked about before. Remember, we get point mutations. You can get frameshift mutations. Okay? Even here, you can get transposition. You get movement of chromosomes. Genes can be moved around, causing jumping genes, etc. After that, after the messenger RNA is made, okay, you can have other situations which occur that can alter the translation process into the end product of a protein. Okay, we'll look at some of these in future lectures, but I just wanted to let you know that there are lots of places in this process of transcription and translation where mistakes can be made, alterations can be made, where the process might fall apart, or you might get a different response during different times of an organism's life. Lots of situations can occur, okay? So we're going to go through some of this expression piece now, okay? So gene expression is really looking at the central dogma, okay? So remember that central dogma theory in the sense that we go from DNA to RNA to protein. And I said before that it's the same in every single organism. Whether you're prokaryote or eukaryote, it's the exact same. So the components, the processes, the products of replication, transcription, and translation is very, very similar between prokaryotes and eukaryotes. Now, that being said, there is some alterations, some intermediate steps that are missing from the different groups. For example, eukaryotes have what we call introns and exons. So after you transcribe your DNA into a messenger RNA strand, before it makes it to what we call a messenger RNA strand, we call it a intermediate RNA strand. And that's because introns are still there. Regions that don't code for anything are still in the messenger RNA. You have to cut those out. This only occurs in eukaryotes. Prokaryotes, this is not the case. They do not have intron regions. So even though the components and really the whole entire process is nearly identical between prokaryotes and eukaryotes, there are some minor differences that I want to talk about when it comes to gene expression. So here you can see the replication process, the transcription process, the translation process, pretty much identical. Right? You're using, helicase to unwind the DNA to create that replication fork. You're using ligase to reform the Okazaki fragments. DNA polymerase III is laying down new nucleotides. So you've copied your DNA. Okay? Let's say you're in need of certain proteins. So then the transcription process can work. You can choose a single section of your DNA, which we call a gene. RNA polymerase will come in, it'll unwind, it'll make a copy of a small section of DNA into RNA. Okay? You then use that messenger RNA to lay down the correct amino acid sequence to form a polypeptide chain or a primary protein. Okay? This is all very similar between prokaryotes and eukaryotes. There are some differences, some intermediates that happen in between these different phases that I'm going to talk about. All right. The one thing about the prokaryotic gene or the prokaryotic chromosome, remember prokaryotes have a single chromosome and it's a circular chromosome. Okay, so it's not linear chromosomes, it's one big circular chromosome. The other thing that prokaryotes got going for them is that their genes are uninterrupted. So when I talk about the human genome and I say the human genome is 3 billion base pairs, we have roughly 25,000 genes, maybe each gene is 1,000 base pairs or something like that. Right? That leaves a lot of DNA not accounted for. Right? Eukaryotes have introns, we have regions of DNA that don't code for anything. They're really thought of kind of as like placeholders in the genome. Okay, so you have chromosomes and you have regions where, there's a gene, but outside that gene, you have large sections of DNA that's not coding, non-coding regions. Okay. Prokaryotes do not have that. It's gene to gene to gene to gene across the entire chromosome. Okay? And so it's uninterrupted sections of genes, which makes it so prokaryotes can copy multiple genes at the same time. Okay? They can also express, so while they're copying the gene, they can also make proteins off of that messenger RNA strand at the exact same time because they don't have a nucleus. Okay? And so they can encounter ribosomes right there next to the single chromosome, and the messenger RNA strand can move right into the ribosome and make a corresponding protein right away. Okay? This makes for extremely fast gene expression. Okay? So you can go really fast. You can copy your DNA and you can express your genes right away. Eukaryotes, on the other hand, okay, it's interrupted. The DNA is. So not only do you have sections of your chromosome which are not genes, okay, you also have sections of your chromosomes which we call genes that have regions that don't code for things. And those are called introns, or non-coding portions of the DNA. The exons are the coding portions of the DNA. And so we're going to look at how do we go about, as being eukaryotes, how do we go about removing these introns and splicing the exons together? Okay. So first of all, that DNA sequence, that DNA gene, first becomes transcribed and it forms what we call a primary RNA transcript. Okay, so this primary RNA transcript is a sequence that is basically the RNA equivalent to the entire gene. Okay. From there, this would include, the introns and the exons. Okay, so the entire gene introns, exons, and this is going to occur inside the nucleus. Okay. Then we have a series of enzymes called SNRPs and spliceomes, okay, that are going to come in and cut out the introns. Okay. So they remove all the non-coding portions and stick all the exons together. So the messenger RNA sequence, it starts off as an entire copy of the gene. And after all the introns have been removed, it's a really small sequence, which we call messenger RNA. Okay? And in a lot of eukaryotes, it's around 90% of the gene is actually introns. So it's like a 10% exon region. But it occurs in these little splices, these little teeny small segments of exons interspliced in between introns. Introns have to be removed, exons all slammed together into one code. Okay, and so this, what I was talking about before is that sequence, that gene could be, 90% introns, non-translated genetic material. After that's all done, and you've removed all the introns, and you've spliced all the exons together, then we put what we call a 5' cap on and a poly-A tail. So you cap the ends of this messenger RNA so it doesn't unravel, so it's protected. Okay? That 5' cap is important because that 5' cap is recognized by the ribosome and the ribosomal RNA. Okay, so here you can see what I'm talking about. So you've made a copy of your entire gene, okay? And you have a bunch of intron regions in here. Okay? These are non-coding regions. They need to be removed. Okay? So they're cut out, and the exon regions are all spliced together. A cap, a 5' cap is added, and a poly-A tail is added. Poly-A just means a bunch of adenines are added to the end. Okay? And now you have a mature mRNA transcript. This is what can leave the nucleus. This is what's going to encounter the ribosomes. Okay? This molecule, not this one up here. Okay? So let's look at this in video form.
Most eukaryotic genes are composed of numerous short coding sequences called exons, interspersed between long stretches of non-coding sequences called introns. The initial messenger RNA molecule copied from a gene includes introns as well as exons. Before this RNA molecule can be used to make a protein, the introns must be removed by a process called RNA splicing. A structure called a spliceosome plays a key role in RNA splicing. The spliceosome is composed of several small molecules called snRNPs, or SNRPs. SNRPs are made up of protein and small RNA molecules. Formation of the spliceosome begins when one kind of SNRP binds to the 5' end of an intron, and a different kind binds to the 3' end of an intron. Additional SNRPs then interact with the complex, bringing the two ends of the intron together and causing the intron to form a loop. This also gathers all of the SNRPs together as the completed spliceosome. The intron is then cut out and the exons are joined or spliced together. The spliceosome then falls apart as the SNRPs are released.
Okay, so there you can see how introns are removed. Right? And in the case of humans, we know a lot about our DNA. We know our entire genome. We know how much of it's introns, how much of it's exons. Okay, and really, in a lot of our genes, 90% of the gene is introns. So this has to remove, this spliceosome complex has to remove a huge amount of the intron regions of that copied messenger RNA. Okay. Now here's the kicker. And this is the interesting piece, and this is the piece that a lot of research is going into to figure out how this works is sometimes when those spliceosomes come in and they remove those introns, they also change the arrangement of the exons. So in other words, you might have one gene that occurs on a chromosome, but depending on how it is spliced together, it can give you a different reading frame, a different amino acid sequence. Okay? So it means that we can get different proteins from the same gene, from the same exon region. You can get different proteins. And this alleviates some of the issues that we had when we came up with the human genome. So when we discovered the human genome, we knew it was 3 billion base pairs long, okay? But we were expecting to find lots of genes, far more than we found, because we already knew that there was at least 100,000 proteins had been described. And so we were expecting to find, well, 100,000 protein, or 100,000 genes, one gene per protein. But that's not what we discovered. We actually discovered that we only have 25,000 genes. Okay? And now what we found out is, due to alternative splicing, we can make about 120,000 different messenger RNAs. Just because the gene itself might code for one exon sequence, it doesn't mean that alternative splicing can't change that exon sequence and give you a variation of that protein. Okay? And we're just beginning to understand this. Okay? So we have a lot of work to do in gene expression front of eukaryotes because of this alternative splicing. Prokaryotes, okay, on the other hand, like I said before, the gene can be translated at the same time that it's transcribed, giving them a distinct advantage when it comes to construction of proteins, at least in a manner of speed. They have a distinct advantage. So again, like I said before, eukaryotes can't do this because, well, you have a nucleus, and you have to have transcription, and then you have to have that original RNA code being the introns being removed, spliced out to form a mature messenger RNA. Then that messenger RNA can leave the nuclear envelope and encounter ribosomes. So in the case of prokaryotes, it all can happen at once. In the case of eukaryotes, it has to happen in stages. Some of it occurs in the nucleus, and some of it occurs in the cytoplasm. Okay? And here's just kind of an overview of exactly what we're talking about. This is showing a eukaryote transcription and translation. So again, there's an enzyme or a protein that your body needs to make. And so that gene is encountered, okay, and RNA polymerase finds the promoter region on that gene, okay, and starts copying it, 5' to 3'. Okay. A new strand, a RNA copy of the entire gene is made. For the majority of genes, there's a large section of introns that need to be spliced out. So the introns are removed by spliceosomes. The exons are spliced together, and you develop a mature messenger RNA strand with no introns, okay, just exons. That material, that messenger RNA, can then leave through the nuclear pore, and it encounters ribosomes in the cytoplasm. The 5' cap will bind to the small subunit of the ribosome in the region of the ribosomal RNA. So it recognizes it, so it can be held on. After that binding happens, the large subunit of the ribosome binds on top, okay? And translation can start to occur, where you're starting with methionine with the start code, okay? And you're moving through until you reach a stop code, adding a single amino acid per 3 nucleotides or per codon through this process. Okay? And like I said before, some amino acid chains can be 300, 500, 1,000 amino acids long, so it can take a lot of transfer RNAs to build those large proteins like that, large polypeptide chains. Okay?
Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Because eukaryotic cells possess a nucleus, their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein.
Okay, so you can see from that video that, the two are fairly different when it comes to gene expression, at least in the sense that prokaryotes can express multiple genes at the same time, building multiple proteins at the exact same time, while eukaryotes, because of the nuclear envelope, the process of intron exons, and the fact that we have linear chromosomes with genes that are separated by regions that don't code, we normally express a single protein at a time per messenger RNA strand. Now, with that, next time I'm going to lecture on how prokaryotes and eukaryotes can alter this situation. How can they shut down the system? What happens if you produce too much messenger RNA? Or what if you have too many proteins in solution? How do you have a negative feedback? How can you stop this process from occurring? Okay? And we'll come back and we'll talk about control mechanisms, not just stopping it, but also starting it, control mechanisms of prokaryotes and eukaryotes next time.